Super. Okay, then I will go. So thanks a lot. Really happy to be here in this today. So I'm Felix. I'm working on EEG and dance in Giacomo Novembre's lab in Rome. And the question of this flash talk is basically how can we capture the complex neural encoding of dyadic dance or collective dance if we're even more ambitious, which raises many challenges, as you may know if you've worked with EEG. One is that when we dance together, basically the brain must encode multimodal information, like the music that one listens to, the movements that I generate, the movements that my partner is generating, and also how I'm going to try to harmonize all of this together to make something coherent. And to address this question, we recorded the dual EEG and 3D full body kinematics simultaneously from dyads of participants, lay persons dancing spontaneously to music. And in a 2x2 design, we manipulated both whether they could see each other and also whether participants would listen to the same or different songs which were played through earphones. And the very cool thing about this setup is that we can know exactly the acoustic signal that the participant listened to. We know exactly the movement signal that the participant generates, and we also know exactly the movement signal that the participant's partner is generating. So we can start asking this question, how are all these signals encoded all together in the brain? And we're trying to, like, one cool way now to answer this question is to use linear models such as temporal response functions that have been put forward by Cross and colleagues. And basically what these models do is that they can be trained to predict the actual EEG activity from the participants in a multivariate manner from the stimulus or the movement regresses. And so you can say then evaluate whether acoustics predict the EEG activity, whether movement signals are associated with EEG modulations, etc. And then you can even look at these little weights here that basically represent the modeled EEG responses. And so in acoustics, in the field, it has been used now quite a lot. And so it's been shown to work very well to estimate auditory responses to speech, music. But with movements, it's been way less used. And so it made us face a very simple question, which is first, what feature can we use? What variable can we use as a regressor? Because we have 66 variables. Like when we dance, we use full body kinematics. So it's a huge number of potential variables. And we did a bit of preprocessing using PCA. That's where you will say if you hear something or not. But basically, for this, you can see the richness of the movement and maybe hear the stimuli. And this complex data, PCA told us that you could reduce it to a set of 15 principal components, principal movements, which basically represent the complexity of the dance movements across participants. And so this helps us to go from 66 to 15 variables, but then we can even reduce further, because what we are interested in is in the principal movements that are associated with EEG modulations. Maybe not all of them are. And so that's what we found in this pre-processing step, that only 1 PM out of these 15 was explaining EEG activity in the participants. And interestingly, its bounce movement is reminiscent of bounce movements. And what this graph more specifically says, if you look at the self and other responses in the brain, respectively yellow and red and yellow, sorry, it means that my bounce movement predicts very well my EEG activity in the center of the scalp, but also the bounce movement of my partner. When I can see my partner, explains very well my EEG too in the occipital part of the EEG. So then when we see this, we say, okay, we can use only one time series now in the model, which is bounce, the time series of bounce. And so we have a simpler model where now we're trying to predict the brain activity from acoustics, my bounce movements, and the bounce movements of my partner. And then because it's a social interactive context, we included a little cherry on top, interpersonal synchrony measure. Very simple. It was categorical, just saying basically, are we going in phase or anti-phase, which means, am I going up and you're going up, down and I'm going down, or as opposed to I'm going down, you're going up. And so then we predict the brain activity, estimate the model brain response for each experimental condition, and we see very interesting stuff because the acoustics predict the brain activity very similarly across condition, which seems reasonable because participants listen to the same music across all conditions. Topographies, maybe we can discuss a bit later, but are mostly frontotemporal, and the temporal profile of the brain response that is modeled is really reminiscent of a PNP ERP, So this, I think it makes a lot of sense. And then we have something similar with the motion self, which is basically predicting the EEG activity. Also similarly across conditions, also makes sense because participants moved in the similar manner, whether they see the other or not. And this is more central in the scalp. And then we could discuss what are these shapes, what are these brain responses, because it's very hard to know, maybe related to readiness potentials, for example. But then the one that is super telling is motion other, because movements of my partner explained my brain activity, but only in the experimental conditions where I could see the partner, which just confirms that the response we have here is really physiologically grounded. And other points that confirm this statement are that the topography is more occipital in that response. And also the temporal profile is very reminiscent of visual evoked potentials when one is watching movement, because we have this negativity at 170 millisecond post onset. And then I don't have time to go into synchrony, but just showing that, again, it might be that synchrony here, the synchrony component is capturing something really interpersonal because it's appearing only in the condition where participants could see each other. And so we'll end an open discussion on the fact that these techniques and hyperscanning, kinematics, and TRFs are super promising for disentangling multiple EEG responses that are ongoing simultaneously yet are distinct in collective dance. And I will thank my co-authors and thank you for your attention.
Thank you so much. That's fantastic. Do you want to maybe go back to your last results slide in case people need to refer back to that? I don't know if there's a question in the room. There's a lot going on. So Peter, is there? Yeah, go ahead.
So just think about the choice of just picking out the bounce.
Can you hear Felix? Is it loud enough for you?
Yes.
Yep, go ahead.
Because you pick just the bounce because that's the movement feature that seems to predict most of the EEG activity. Does predicting most of the EEG activity mean that it's the most important to look at? Or is it just because it's like a big body movement? I was wondering, like, does it also fit the people's experience? Is that also what they care about if you would ask them? Not that you necessarily ask them, but I'm just wondering about the truth.
I just missed maybe one part of the question, so don't hesitate to come back if I didn't get everything. The answer is that, yeah, we use this preprocessing really tied to the specific question, which was predicting the brain activity, EEG activity. So basically we want to find EEG modulations that are associated with movement that you see or you generate. And just by seeing that, for example, another type of movement, such as, let's go back, such as side sway, this movement did not elicit any prediction in the brain, means that we could not capture, does not mean there is no EEG response related to side sway, but in our context, we could not capture activity. So that was very basic, was just to say then, why would you put it then to train a huge model that takes days to be trained and then show only results on one. But also to add a bit of meat here, I want to say, like, if there is no response here captured by our model, is also, of course, probably correlated with the signal to noise ratio, because bounce was a movement that was really that was done at the beat a lot of times. Side sway was more slow. So this is kind of also potentially just a limitation of the setup and the technique. It does not mean that it's not all of the other ones are not important for social interaction in that case. And when you're interested in more like kinematic and behavior, you could see that they have actually an important role. And we've also seen that in another study that Giacomo is going to present this afternoon. So of course, they are also important in the interaction if it answers the question.
Really exciting.
Yeah, I would just add that I think that what we'll probably find, just linking things back to Emily's talk earlier today, is that as a function of expertise, you're almost certainly going to be incorporating more of these socially relevant or temporally relevant cues. And I think what's so interesting about your results is that combination between the acoustics channel and the visual channel, because assumedly also as a function either of expertise or accessibility, like how much you can see, whether you're in an environment where you can't necessarily see everything and track everything, maybe you'll do so by the acoustic channel. So I think there's a lot of richness in what you're doing, but also it's an extremely complex thing to try and capture. Again, just to reiterate that we'll be doing a panel discussion on this idea of like how to best capture. And I think what you've tried to do so neatly, and I'd like to commend you on also the structure of your talk, which makes super straightforward for us to break down this multi-sensory, the multi-sensory components of how we coordinate with others, as well as the social aspect. So I really liked this juxtaposition between, obviously you can synchronize at a very self-related level, right? You can just try and maintain a steady tempo and ignore the other person, but you've captured that they're both brain and behavioral aspects that allow us to do so more socially. So thank you so much. I don't know if there's another question in the room. Yep, go ahead, Julia.
Lovely to see you again, Felix. Amazing talk. I was just wondering what EEG system you used for the data collection.
Yeah, that's a good question. Because we used a Biosemi 64, and the thing we did to be able to apply it to dance was to order 2-meter long cables that we would hang. Of course, we did not allow them to go, in kind of crazy dance where you could rotate and go in the middle of the room. So this was like controlled in terms of you have a space of one, like let's say 1 meter square, and then you're facing each other and you dance a bit like in a disco, but not going all around. But this was also cool to control a bit the head movement and everything because if we go crazy wild for the first experiment, then maybe we will have just had non-usable data. But so yeah, it was okay to have this EEG. Because then we had very precise and 64 electrodes and still could hang, not to bother the participants. But of course, they could not have rotated or done, I don't know, breakdance. This would have been tricky.
Yeah. Anybody else in the room? Otherwise, I will just thank you again, Felix, for your great talk, for your amazing initiative. This is no easy task. And I'll just suggest that if anybody in the room or in the bigger, wider world web who's joining us on YouTube, if they have questions, they might pop them into the YouTube live stream chat, Felix. So do keep an eye out there. Yeah. Otherwise, just thank you one more time for your great talk.
Thank you so much.